Namaste students, Khush raho betiya. I JVN Dr. Chandrakala, Assistant Professor Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the synthesis of cell wall. Okay? So the synthesis of chitin is mediated by specialized organelles or cytosomes. Okay? So which is an inactive chitin synthesize are delivered to the apical plasma membrane and become activated upon a contact with that of a lipid bilayer. Okay. So the microvessels visible especially in the core region of the likely to be ultrastructure and manifestation of the cytosomes. Okay. In contrast, the structural protein and the enzymes travels together in the larger secretory vesicles and are discharged into the environment when the vesicles fused with the plasma membrane. Okay, so here the most of the proteins are fully functional by the time they transverse the plasma membrane. Okay, so the glucose are secreted by the secretory vessels as partly form the precursors and undergo further polymerization in the nascent cell wall. Okay, they are synthesized entirely at the plasma membrane. So the cross link of the glucons with another components of the cell wall take place after the exclusion into the cell wall. Okay, so here it provides the individual linear or beta-1,4-N acetyl glucosamine chain excludes from the plasma membrane are capable of undergoing self-assemble into the chitin microfibrils, but these are the subjects to the certain delay during which the cross-linking with the glucon must occur. Okay, so here the glucon in turn becomes alkaline insoluble only after they have become covalently linked into the chitin. Okay, once the structure folds into a place, the wall matrix can be assembled. Okay, so the softness of the apical cell wall is also influenced by the activity of the cell lytic enzymes such as kinase or glucanase. Okay, further when the certain mycota grow under the condition of the hyperosmotic stress, this cell wall is measurably softer during the secretion of the endo-1,4-beta gluconase. Okay, so thus permitting continuous growth when the turgor pressure of the is reduced or even absent. Okay, so since higher mu mycota, both the cell wall material and the synthetic as well as lytic enzymes secreted together by the vesicles, it appears as a portion of the movements which influence the direction or the speed of the apical growth directly. Okay. So of course the cell lytic enzymes are also necessary for the formation of the fibril branches and which usually arises by the localized weakening of the mature fully polymerized cell wall. Okay. So the endo-beta-1,4 glucanase has been shown to involve in softening the mature region of the hyphae in the growing strips of the of different like protein bodies or permitting the intercalary hyphae of the extension. Okay. So the property of the cell wall depend on many types of the environment in which the hyphae grow. Okay. And another difference between the submerged of the aerial hyphae is caused by the hydrophobins, okay, which are the structural cell wall protein with a specialized function in the physiology or morphogenesis or pathogenesis. Okay. Some hydrophobins are constitutionally secreted by the hyphal aping. In submerged culture, they diffuse into the medium and as a monomer, whereas they polymerized by hydrophobic interaction on the surface of the hyphae exposed to the air, thereby effectively rendering the hydrophobic on the surface of the hyphae or the spores in a flow. So some fungi are wall-less during the assimilation stage of the life cycle, and this is the true especially of the certain plant pathogens such as the plasmodiumorphota or insect of the pathogens. Okay. So here the motile spores or the zoospore of certain group of the fungi swims freely in the water and bursting due to the osmotic inward movement of the water in preventing by constant activity of the water exposure vacuum. Okay. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, please write down in the comment box. We'll resolve it in the problem solving session.